The next set of screencasts are taken from Muscle Physiology 2 lecture, and we're going to focus on cardiac and smooth muscle and maybe compare and contrast those with the information that we've given you on skeletal muscle in screencasts 1, 2 and 3. So this is officially screencast 4, and what we're going to do in this and the next 1 or 2 screencasts is to let you understand cardiac muscle function, and in particular to describe excitation contraction coupling at cardiac muscle and highlight maybe the different proteins or the similar proteins and the different proteins that are involved in EC coupling in cardiac versus skeletal muscle. We'll then go on to talk about smooth muscle function and EC coupling at smooth muscle, and the lecture aims overall, I guess, are to be able to compare and contrast contractile cell function. So to know the differences between skeletal muscle, cardiac muscle and smooth muscle, in particularly in terms of how they're involved in excitation contraction coupling. So this is really a couple of placeholder slides. I put this slide up back in screencast 1. Hopefully now we know a little bit about skeletal muscle and its properties. Hopefully after the next couple of screencasts we'll know similar things about cardiac muscle and smooth muscle. So again, maybe a placeholder slide, hopefully that you'll be able to identify this contractile cell, this muscle type as, I'll just pause, cardiac muscle. Okay, so this is what cardiac muscle looks like, and hopefully you can differentiate between this and skeletal muscle and smooth muscle, initially in terms of morphology, appearance, but also hopefully after the next few slides in terms of organization and function. So maybe again, just to compare and contrast some of the properties of cardiac muscle. Cardiac muscle forms what we call a myocardium, and we'll see what a myocardium is. It's a joint series of cells that amalgamate together in order to observe regular synchronous contraction of cardiac muscle. It's involuntary, so it's not neurogenic. Remember, it's not subject to control by nerve input. We're not thinking about how to control our cardiac muscle, our myocardium. It's done by the autonomic nervous system. Innervated by the autonomic nervous systems, it says here. Cardiac muscle is controlled as well by a number of what we call intrinsic physiological factors. One of these most important factors is the amount of blood returning to the heart. So this is the venous return to the right atrium of the heart. A lot of the diagrams that I'm going to give you over the next couple of slides I've shared from lectures from Alistair McNeish, who also tells you something about the cardiovascular system. I'm going to focus on the muscle side of it, but you'll see that several of the slides are shared. And of course, when we ask you questions about cardiac muscle, obviously feel free to bring in information from maybe the lectures on the nervous system, but also the lectures that Dr. McNeish is going to give you on the cardiovascular system, because they're very much shared with each other. Hormones are also important for regulating cardiac muscle. Cardiac muscle has a number of similarities to skeletal muscle, so it's got this striped, striated appearance, and you'll see that when we go through the contractile process, the EC coupling, there are a number of similarities. In terms of morphology, it's branched and connected by gap junctions. So this is an important difference because we want synchronous beating of muscle. It's one of the few situations in the heart where we use, in the body, where we use gap junctions instead of chemical synapses. I think this is a diagram from one of Dr. McNeish's lectures. Maybe just to point out here, we have these important membranes that we mentioned probably when we were talking about individual muscle fibers, muscle as an organ, how it's made up. Probably the take-home messages here is we've got an epicardium and an endocardium, the outer and the inner layer surrounding the heart. Most importantly, in terms of contraction, is the myocardium that we mentioned before. So the myocardium is a muscle layer connected by gap junctions and made up of these individual cells called cardiac myocytes. And again, this is where it fits into this diagram here. Again, maybe some structural terms that we need to relate to function. So there are a number of nodes which control the electrical activity of the heart. The sinal atrial node, hopefully you'll know this better as the pacemaker, and you'll recognise this term. There are some internodal pathways between the SA node and the AV node down here. So the atrioventricular node, the AV node. Number of nerve fibers, bundles of His, bundle branches, and Purkinje fibers, specialist nerve fibers within the heart. Again, this relates a little bit to the way that the heart is under some neural control. Remember, the heart conduction itself is myogenic, not neurogenic, but there are important fibers present in the heart that we probably need to know about. Yeah, again, maybe a slide taken from Dr. McNeish's lectures. Just this really, the reason that I've shown you this is to show you this syncytium or connection of contractile cells. So this gives you the idea that the myocytes are multinuclei, that they branch, and that they're slightly different to the organization of skeletal muscle and smooth muscle. We can see some ideas here about nerve activity, and again we'll talk about these over the next few slides. Essentially, the take-home message from this is that the reason that the morphology of the cells is important is because it allows depolarization to spread across this syncytium using these gap junction connections. Slightly different, well, different to what we see in skeletal muscle and smooth muscle. So yeah, this is a slide maybe just to focus a little bit on the difference between the nerve action potential, which hopefully you remember from the previous set of screencasts when we talked about the nervous system, and the cardiac action potential. So what we see is in different places throughout the heart, there are different types of action potentials, if you like. This one is a pacemaker potential, and these two here, particularly the Purkinje fiber and the ventricular muscle action potential, are most characteristic of what we call the cardiac action potential. So what we can hopefully see here, in comparison to the nerve action potential, which has a very rapid upstroke due to sodium influx, and a very rapid decay over about the course of a millisecond due to potassium channels opening, is that we get this elongated plateau phase. And essentially what we'll see on the next slide is that this elongated plateau phase is due not to entry of sodium in this rapid upstroke or efflux of potassium in this downstroke, this repolarization or hyperpolarization phase, but is due to the opening of voltage gated calcium channels. So the main difference between the nerve action potential and the cardiac action potential, typified probably by Purkinje fibers here, to a certain extent to the ventricular muscle action potential, is this elongated phase due to the opening of a specific type of calcium channel called an L-type calcium channel. This information here just really relates to the fact that these potentials allow us to get spontaneous action of the heart in this synchronous manner because of the syncytium that we showed you on the previous slide, because of the connections with gap junctions. So yeah, let's just focus a little bit on this just to reinforce what I've been saying. So that as with the nerve action potential, we get sodium influx. Yeah, okay, the diagram, the arrow is I guess is showing sodium coming into the cell. Remember that this is minus 70, this is up to about plus 30. So this is the inside of the cell. Sodium coming in makes it more depolarized. And similarly, let's just look at phase 3, because that's the same in the nerve as well. We get potassium efflux, and we get this balance through the sodium potassium ATPase. The important take-home message here probably is that the plateau phase that we showed you on the previous slide is due to calcium influx through what we call L-type calcium channels. And just to focus on here a little bit, we saw the term DHP, dihydropyridine receptor, and actually dihydropyridine receptors and L-type channels are one and the same. It depends whether you're looking at it from a let's say an electrophysiological point of view, where we'd call it a voltage dependent calcium channel or a voltage gated calcium channel. If we're talking about it from a biochemical point of view, some textbooks will call this a dihydropyridine receptor. Basically, dihydropyridines bind to these L-type calcium channels. So dihydropyridine and L-type, VDCC or VGCC, are synonymous terms. They all mean the same thing. So let's just spend a little bit of time considering excitation contraction coupling in cardiac muscles and some of the structures that are important for function. So myocardial cells contain actin and myosin, the same way as we described for skeletal muscle, and they also contain T tubules. What is slightly different is, remember we talked about before about red and white muscle fibers. In skeletal muscle, cardiac muscle obtains oxidative energy from aerobic metabolism. So they're high in mitochondria, provide energy, use aerobic metabolism. At least 60% of energy is derived from the metabolism of fats, around about 35% from carbohydrates and 5% from amino acids and ketones. So compare and contrast this with the different fibers that we use in skeletal muscle contraction. Yeah, I think I'll just basically take this screencast to talk about cardiac muscle, and then we can use the other screencast to talk about smooth muscle. So excitation contraction coupling in cardiac muscle, you're going to see there are a number of similarities, a few minor differences between EC coupling in skeletal muscle and cardiac muscle. So we talked about this phase 2 of the cardiac action potential. This is when depolarization, this time of the myocardia, allows voltage gated, voltage dependent calcium, L-type calcium channels to open and sodium to come in. Calcium to come in. Again, everything's triggered by calcium. So calcium is sensed by ryanodine receptors, which are also present in skeletal muscle, and it triggers what we call calcium-induced calcium released, CICR, as the final trigger for excitation contraction coupling. This occurs to a certain extent also in skeletal muscle. Again, look back at your notes. The proteins that are important, again, as with skeletal muscle, are troponin. Calcium interacts with a number of subtypes of troponin in the cardiac excitation contraction coupling. So troponin C, troponin I, and another one called troponin T, which we'll show you on the next slide. As with the sliding filament model that we showed you for skeletal muscle, this interaction uncovers the myosin binding site on actin, and cardiac muscles contract according, again, to the sliding filament model. Intracellular calcium is reabsorbed, again, using this protein that we mentioned before in skeletal muscle, the SERCA pump. So the sarco-endoplasmic reticulum calcium ATPase. We also, in the heart, use a sodium calcium exchanger and an ATP dependent calcium pump. Probably again, as described for skeletal muscle, as intracellular calcium decreases, calcium dissociates from this troponin C, and the binding site on actin is inhibited. So look back again at that sliding filament model. It pertains equally to the cardiac system as it does to skeletal muscle. And ATP, again, look back at the sliding filament model. The way that ATP is converted between ADP and inorganic phosphate and then reattaches is the same for EC coupling in cardiac muscle as it is for skeletal muscle. So let's just go through a few diagrams to show you that in a little bit of detail. So first of all, calcium enters, or the cardiac action potential, which you can see here, causes depolarization. This is the T-tubule. This is our sarcolemma. Calcium enters down its concentration gradient in this L-type calcium channel. Again, remember when we looked at skeletal muscle, this was probably labeled as a DHP receptor. It's the same thing. Calcium, get this calcium-induced calcium release, CICR, due to the action, due to the role of this ryanodine receptor, which allows calcium to be released from these SR stores. Calcium interacts with these different forms of troponin, so TNT, C and I, essentially calcium-driven contraction due to the binding of calcium of troponin subtypes in cardiac myocytes. Circa, this pump that we talked about before, ATP-dependent pump. The reason we need ATP is because calcium is pumped against its concentration gradient back into these terminal stores, the SR stores. And again, a couple of other proteins are important. Sodium calcium exchangers, NCX, exchange the NCX changes sodium and calcium, and we also have our sodium potassium exchanger that we use in other systems to maintain the gradient of sodium and potassium inside and outside the cell. Calcium is removed by this exchanger and pumped into the internal stores, as I said, using this Circa pump. Yeah, let's just complete cardiac muscle before we stop the screencast. If you need a break, just pause the screencast. This is really just to show you, before we mention these two periods, the absolute refractory period and the relative refractory period. And the reason I've put this on here is really just to show you that because of this plateau phase, the absolute refractory period for cardiac muscle is much wider. And if you remember before, I said that this is the period where you absolutely cannot generate another action potential. And the reason that it's wider in cardiac muscle from a physiological point of view is for safety. So what we don't want is for the heartbeat to be irregular, and regularity of heartbeat is essentially dictated by the fact that this ARP is longer. You cannot start another action potential until the relative refractory period due to this plateau phase. So it's kind of a safety factor that prevents the heart from firing irregularly, which of course would be a bad thing. Yeah, ventricular muscle contraction is completed shortly after the relative refractory period. So the next muscle contraction is triggered regularly once this period finishes. Okay, so we spoke for about 20 minutes. What we do is we'll finish this screencast here, and I think the final screencast will be again on smooth muscle and comparing and contrasting that with cardiac and skeletal muscle. So we'll take a break here.